Good afternoon and welcome to the Chamber of Medford and Jackson County's virtual forum. Today's program is sponsored by Providence Health and Services and brought to you by our season sponsor, the Cow Creek Tribe. I'm Dr. Tom Lorish, Chief Executive of the Southern Oregon Service Area for Providence Health and Services in Oregon, and I will be emceeing today's program. Also with us today, we have Chris Pizzai, Chief Executive Officer at Providence Medford Medical Center and Chamber Board Member, who will be helping to answer questions during Q&A at the end of the program. Today's keynote speaker is Dr. Walter Erba, MD, from the Providence Cancer Institute, who will present on COVID research in the state of Oregon. Thank you for being here. I would now like to take the time to thank all of the forum sponsors who help us put on these programs. Thank you for your continued support. The Chamber is grateful to bring everyone together on topics that are important to our business community. Just as we do for every virtual forum, I would like to remind you of how today's program will work. If you've attended one of these programs, you already know this information, but for those of you who are new, I'd like to explain a few things. As you've noticed, you are not on screen and do not have video or microphone capabilities. You are simply an attendee. There is a question and answer feature of this webinar. Please find at the bottom of your screen an icon with Q&A on it. Here you will be able to type in your questions for our panelists. Please feel free to type in your question as they come up during the presentation. Just know that we will be answering them at the end of the program. I will be reading through your questions and asking a few at the end of this presentation. When it comes to healthcare, it goes without saying that this last year has been unprecedented. Providing healthcare during a pandemic certainly brings challenges, but it also brings opportunities for us to learn and grow together. I have been humbled and motivated by the extraordinary efforts from our caregivers and providers. They are an example of dedication and perseverance like I have never seen before, exemplifying the Providence vision of health for a better world. Our focus on health goes beyond the clinics and hospitals. Providence values science and innovation in healthcare, and that brings us to our speaker today, Dr. Walter Erba. Board certified in oncology and internal medicine, Dr. Erba specializes in clinical research and in the care of patients with melanoma. He has 30 years of experience in the development of immunotherapy. Dr. Erba earned his PhD in immunology at the UCLA School of Medicine and his medical degree at the University of Miami. Dr. Erba completed his residency at Morristown Memorial Hospital in New Jersey. He has earned several prestigious fellowships focused on medical oncology research and development. In 1993, Dr. Erba joined Providence Health and Services in Portland to lead the Robert W. Franz Cancer Research Center in the Earl Child's Research Institute at Providence Cancer Center. He has served on several oversight committees and advisory boards focused on cancer immunology and immunotherapy. He also serves on the editorial boards of the Journal of Immunotherapy, Journal of Translational Medicine, The Oncologist, and the Journal of Immunotherapy of Cancer. During the COVID-19 pandemic, Dr. Erba has led our vaccine development efforts for Providence. He's here today to tell us about this important work. Please welcome Dr. Walter Erba.
Thank you very much, Dr. Lorish, and thank you all for attending today's meeting. I'm going to try and share my screen, and this is always the hard part. I am screen sharing because my slides are not showing up. So give me a second while I work through the screen sharing component here. And share. Nope. Stop share. Let me try again. I apologize. None of my files are showing up. I will be talking today about a bit about coronavirus, bringing people up to date where we are in our management of patients with the coronavirus. I want to talk about the research that we have been conducting as a nation, but also the work that we have participated in as Providence for trying to develop new therapies for patients infected with the coronavirus. And I want to finish with discussions about the vaccines that have been developed across the country and that are now available for our use. And I'm sure many of you have been recipients of by now. I'm hoping you can see my screen now. There's the title of today's talk, and where I wanted to start was a couple snapshots. This is a map that perhaps many of you have seen many times over the past year. This is almost exactly 1 year ago, April 13th, 2020. This is a Johns Hopkins map where they were keeping track of all of the new coronavirus cases throughout the world. And you can see up in the left-hand corner that this is when there were 1.8 million cases in the world. 558,000 of them were found in the US. And interestingly, now that is the number of deaths that we are seeing, have seen in the US since that time. But they're not seeing they're not seeing my slide. I apologize. We're trying to figure out why it worked in practice and is not working right now. Well, the first slide is a map of the world and shows the cases. And I guess I just try and make the point that we've gone from 1.8 million infected people in the world last April, and now we have 132 million people infected in the world. And in the US alone, we've had 31 million people infected by the virus and 556,000 deaths. And the next slide was to try and give you an idea of how this has impacted Oregon, where we've had far fewer cases than other parts of the US, but we have had our challenges. I'm sorry that while I'm talking, we're trying to find the right screen to show up so that I can share these slides, because obviously this would be much more beneficial for.
There you go. There it is, Walter.
Okay. All right. Whatever we did. Thank you so much.
Excellent.
Okay. Thank you. So here's about a week ago. You can see the U.S. is all too infected, where we've had more than 500,000 deaths. When you look at how Oregon was affected, here again is a snapshot of last year. So if you remember, it was the beginning of 2000 that we first started seeing the coronavirus cases, and you can see early in February and then in March. And I will tell you, this looks like a terrible peak as you look into March. And I remember March very well. We were having 7:30 meetings every day across the health system. We were worried about having enough masks, enough personal protective equipment. And if you look over here, the number was 80 cases, and the blue here is who's in the hospital, those that aren't hospitalized, and people that are hospitalized, but we weren't sure if they were viral positive. So take a picture of that, 80. The number was 80. We were all in a panic. ICUs were filling up. We weren't sure we had enough protective equipment. And now here's a curve that shows the whole, pretty much the whole year from March 1st of last year to March 1st of this year. And that you can see over here on the left-hand side, that was the peak on the last slide. And what was 80 is now up to 1,500. And you can see what we lived through with the second wave in July, and then another wave in November and January with peak numbers of 1,500 patients per day infected. And you can see the decline and a suspicion that the cases are increasing. But again, as much more comfortable we are now than we were a year ago, there are a lot more cases around still in the hospital. Right now, there's about 44 patients in Providence hospitals in the state of Oregon. We had 100 in just Providence Portland Medical Center at one time was our peak. So you know how difficult the time was when we had all of these patients in the hospital, and it's no surprise if you look at 2020 that the third leading cause of death in 2020 was the coronavirus infection. Third behind cancer and heart disease. And just briefly, you're now familiar with this probably by watching the news, perhaps by family members or friends, but the signs and symptoms of COVID present like many other illnesses. The flu, for example, people get fever and cough and they get tired and they lose their appetite. We get really worried when patients start complaining of shortness of breath, they get muscle aches. They can have GI symptoms, which can often make it difficult to figure out just what is happening. That was particularly true in the early days. More recently, it's assumed patients have COVID unless proven otherwise. In the beginning, when tests weren't so prevalent and they weren't available to everyone, it was often a challenge as to who would get testing, and these symptoms would help us determine who appeared to be at the highest risk, although these non-specific symptoms are not perfect. And we know we missed lots of patients, and we know that many patients can have can carry the virus and be asymptomatic. A couple things that became sort of pathognomonic or clearly indicative of the presence of coronavirus were loss of smell and loss of taste. I'm sure you've heard that on the news, and that often preceded people developing signs or symptoms that led them to the emergency room or required hospitalization. And those recover, maybe not completely and not quickly, but those are things we were looking for. And when you saw those things, it led to testing. And the picture on the right shows that the original testing was a probe deep into the back of what we call your nasopharynx, which for those of you who've had this test know it can be rather unpleasant. We can now look into what's sputum or spit, and we've learned more about where the virus is present in secretions. So not everyone has to have as deep a sample taken, although that is probably the highest quality sample, the most likely to give the best results. And the ability to detect the virus does depend on the quality of the specimen. Now, the gold standard for the test is to look for genetic material of the virus, and that's by polymerase chain reaction, which is a fancy test that can be done cheaply now. A guy won a Nobel Prize for it 30 or 40 years ago by figuring out that you can make millions of copies of DNA so that you can detect just a couple of viruses in a sample that is taken in the emergency room or in your doctor's offices. So that detects genetic material. The other test that's available is to look for virus antigen or protein. This depends on antibodies. This is what people talk about when they think about having a home test. It's very useful. I mean, if any of you are sports fans, when you hear during the NCAA tournament when they were testing everybody all the time, they were looking for antigen or proteins. The test comes back much quicker. The problem is there's a higher rate of falsely positive tests. So if an athlete tested positive, they had to have a confirmatory test, which was done by looking for genetic material, PCR, and that was the definitive test to decide whether the athlete could play or not. And that's pretty much how we do those tests now. For the COVID pandemic, initially, we didn't have enough tests. We didn't have enough pieces of equipment to even do the testing. So we developed our own test. Our molecular genomics laboratory was amazing. We had amazing philanthropic support who helped us buy the new pieces of equipment that came out over time that took this from being a test that had to be done on a very big machine in a central pathology lab to a test that could be done at point of service, and little pieces of equipment were purchased for places across the system. I don't know exactly how Medford did its testing, but I know that it was possible also to develop testing in Medford so that results were available. So once people developed symptoms, they were then tested, they were positive. How we managed them depended on the severity of their illness. Now, we all know by now that most patients who get it have mild to moderate symptoms. On the order of 80+% of patients fall into that category and can be managed with outpatient treatment. Some of you may have watched Chris Cuomo on CNN. He stayed in his basement but did his TV show every day. So lots of people still feel okay. Others have a bit more severe illness, and they develop what's called dyspnea, which is shortness of breath. Hypoxia is low oxygen concentration, and they can get lung involvement. And this is where people end up being in the hospital. It can become life-threatening. They can develop a critical illness, and the pneumonia can be so severe they get respiratory failure, and their blood pressure can fall, and then multiple organs fail, require very much severe intensive care, lots of hospitalized resources. And you can imagine the nurses and the doctors and the housekeepers who had to help these patients were also being exposed to the virus at that time. And it was a scary time for everybody caring for these patients. A lot of patients developed what we call adult respiratory distress syndrome, which is a very severe illness that can follow lots of different injuries, not just COVID. And early on, among all our hospitalized patients, 1/4 of them were admitted to the ICU, and 3 to 17% developed ARDS. And you can see how severe. This is a chest X-ray, and those lungs should look black with the heart in the middle. But you can just see all of this white, cloudy material. That's virus and our body's immune response and inflammation trying to get rid of the virus and eliminate it. And it causes such an infiltration of both virus and inflammatory cells that you can imagine it's pretty hard to breathe when your lungs look like this. And these are patients that ended up needing to be put on a ventilator and having machines breathe for them. And you also remember there was a time in the crisis early on where we weren't sure we would even have enough ventilators for these patients. Thankfully, in Oregon, it did not come to a time where we had to choose who gets a ventilator and who doesn't, although it did. Friends of mine taking care of patients in Italy did indeed have to make those incredibly difficult decisions. Now, we know that there are certain risk factors that predispose to severe illness. Underlying medical conditions like heart disease, diabetes, chronic lung disease, hypertension, and cancer can make COVID infection worse. And one of the best guides we had was age, right? If under 54 years of age, less than 1% of people had severe illness. And you can see as the age group increased, the percentage of patients who got into trouble, required hospitalization, required ICU care, and died increased. And this is the kind of thing that affected how we brought out vaccines when they became available. Here, You've probably all seen pictures of the coronavirus on TV, and this is a cartoon, and it's the SARS-CoV-2 virus that causes COVID-19 disease. Couple key things to remember because it impacts the research that was done and how we do treatment and what the vaccines recognize. The RNA is the virus's genetic material. That's what we look for and detect. It also happens to be what some of the vaccines are made out of from Pfizer and Moderna. And the spike protein is the other important component because it's key to infection, but it's also the target for all the vaccines. And this is the spike protein. It likes to line up in what's called a trimer. So three of the proteins gather together, and they form this tulip-like structure that recognizes an ACE2 angioconverting enzyme 2 receptor on our cells. And the main cell in our body that expresses this ACE2 are the cells that line our lungs. And so it's no surprise that pneumonia is the big problem. They're also in our nasopharynx. They're in many other parts of our body, but the virus is spread by droplets, inhalation. The virus finds its way to the lungs, finds the ACE2 receptor, binds to the cell, and then starts to cause all of its trouble. Now, I know this is a complicated slide. I'm going to try and explain it simply because to understand or appreciate the research that's been done, it's very helpful to know a little bit about how the virus works and infects our cells. So here in the top left, you can see there's the ACE with the spike protein, and it binds there. Then it fuses with the membrane of our cell, and the virus gets inside, and then it does what it's called uncoating, which just makes a hole that releases its genetic material inside our cells. And then it doesn't have the capacity to reproduce its own genetic material. It relies on our cells to do that reproduction, which makes treatment a challenge because the process that it's using are our own processes, and we can't interfere with our own process for making new DNA, et cetera, because it will cause toxicity and side effects. But so the genetic material gets inside the cell, messages are taken, and these proteins are made, and they make, the first proteins they make are those that help replicate their own genetic material. And then that genetic material is made into new proteins that they need to form a new virus. And then that virus is extruded from the cell in millions of copies to go out there in the bloodstream or wherever and infect other cells. So this is what we're dealing with. And on top of that, we're dealing with our own body's need to recognize and eliminate that virus. And while it's forming an immune response and trying to eradicate the virus, it sometimes gets caught in this huge inflammatory response, which causes a lot of collateral damage. And for a year, we've been working on trying to understand, okay, what's viral damage versus what's collateral damage from an uncontrolled immune response. And so if you look at the boxes that are in these slides, these are all the places where we have been involved with research to try and help patients and interfere with the virus's ability to make us sick. One place you can look at is blocking antibodies. So you've heard of convalescent plasma and monoclonal antibodies. These are, convalescent plasma is, okay, people get SARS-CoV-2, they get better, must get better because they made antibodies against it. So let's take their antibodies and give them to other people. That's the principle of convalescent plasma. And that blocks infection here. Then there are other things that can target these cofactors that the virus needs to get inside the cell. Hydroxychloroquine is something that was supposed to help by blocking the membrane fusion. There are HIV drugs, which include what's called a protease inhibitor that can interfere with HIV replication, but they've been tried in many trials. So far don't seem to work very well against this virus. The one drug that does seem to work is remdesivir, and that seems to be able to inhibit viral replication so that the virus doesn't make as many copies of itself, and thereby we can limit infection. And then there's been a lot of work on combating the inflammatory response that we make. And these are just drugs that we've tried here and others have tried to inhibit inflammation and make decreased hospitalization, improve survival, all the things we need to do for protection of our patients against COVID. And so this is a summary slide of some of the treatment, and it just introduces the idea of prevention. And I'm going to show you some slides of the results of these treatments that we've tried and show you that even in the ones that turn out to have beneficial effects, I hope to show you how minor those beneficial effects are and why it is so important for us to focus on the prevention side of things rather than on treatment. So for the antivirals, the numbers in red here are the number of trials in Providence, St. Joseph Health from Alaska, Oregon, Washington, Montana, California, and Texas. This is how many patients we put on these clinical trials. Remdesivir, 511. And these were, our doctors were authors on the trials and the data that went to the FDA to show benefit. We've tested the anti-inflammatory approach with a number of drugs. I'll show you a little bit of that data. We put more than 1,400 patients treated with convalescent plasma, 244 with monoclonal antibodies. So in total, from all of our Providence sites, we put, now this is a few weeks old, we put more than 3,000 patients on clinical trials. Now, what this means is that these 3,000 patients got drugs they wouldn't otherwise have had access to. Now, not all of them worked, so they didn't all benefit, but some of them did work, and it means that our patients got access to beneficial therapy long before it was available through the FDA. And I think that's an important service that we in research provide by getting patients access to potentially life-saving drugs before they're on the market. These are some slides that show the difference between patients who got remdesivir and placebo. And most of these all these slides show the percent that recover. And what you can see is there's a pretty modest difference between the patients who got placebo and those who got remdesivir. Now, when you do the statistics, these are beneficial differences, and they went to the FDA, they were reviewed, and they have been approved by the FDA. But you can see that there's pretty small differences between the two. Dexamethasone, which is a steroid we've used for years and years and years, and probably is the cheapest medicine available to treat COVID. You can see here, usual care mortality is worse than with dexamethasone, and dexamethasone is the only drug that was shown to lower mortality. But you can also see here that these differences are rather small, right? The benefit, obviously, we all know which curve we would rather be on, or we'd rather have our family member be on. But given the effort and the worldwide effort and the extent of work, this is a pretty modest benefit. Convalescent plasma, minimal to no benefit after thousands of patients being treated. Monoclonal antibodies maybe a little bit better. Have to remember that most of these patients are severely ill, and the idea is maybe the earlier we look at treating patients, the better we're going to do. So this is an active area of investigation. This is another trial that just came out and was approved by the FDA, another drug which is anti-inflammatory. So makes sense, right? If you get a little benefit with an antiviral drug and you can get a little benefit with an anti-inflammatory drug, what happens if you put them both together? And the answer is you get a little bit of a benefit, but not as much as we would all like to see. And you can see here, if you add baricitinib to remdesivir, you recover 1 day sooner. You get a few days benefit in reduced oxygen flow. Now, if you count this against hundreds of thousands of patients being treated, this is significant and will help lower the cost of care, but it's not the lifesaver we would all like to see. And so current drug therapy, which isn't overly satisfactory, consists mainly of remdesivir. Still question of when or if to use convalescent plasma or monoclonal antibodies. Everybody who's sick enough gets dexamethasone, and these other drugs are still being studied. So this is the reason we're all interested in vaccines. The benefit of all the tens of thousands of patients who've been treated with all these different drugs. We don't have a drug that works very well. We have to keep working at it to try and find the Achilles heel of the virus. But there are ways to prevent. And of course, we didn't have any vaccines a year ago, so we had to focus on therapeutic modalities. But now here, a year later, here are the results of two different vaccines. And you can see here with Pfizer and Moderna placebo, the infection rate, and you can see here the low or almost total prevention of infection in patients who got their vaccine. And the timeline here is just to show how amazing this is to have occurred in just under a year. How does it work? The virus message is put in a lipid nanoparticle. It gets injected into your arm, and you're sore because it gets into a muscle cell. The message goes into that normal cell's machinery. They make the S protein, and after the S protein is made, your body makes an immune response against it. Now, I haven't shown, I'm not going to show a lot of the data, but it's safe. Pain at the injection site. Some people get fatigue, headaches, a small price to pay for the benefit that we're receiving. Severe side effects are really, you can see them in both arms, and they're really, with the mRNA vaccines, almost absent. Now, there are other vaccines out there. Most people in the US have gotten Moderna or Pfizer, but the Johnson & Johnson vaccine, which is the same spike protein, but they use a cold virus, an adenovirus, to inject it. That was approved in February. Now, AstraZeneca has another kind of vaccine. It uses a chimpanzee cold virus. It also has the same spike or S protein, and it's been approved in the UK, Argentina, India, but not in the US. And if you've been reading the paper, there have been some questions of unusual side effects with this virus, so I'm not sure how quickly it will be approved here in the US. Now, so there are still some ongoing questions, and maybe you will raise some of these in the question and answer period. But the big question is, how long will this last? Is this going to be like a tetanus shot or one of those other vaccines where you get it and it works for 10 years? Or are we going to have to get it every year or even more often? And this slide just shows antibody responses. And so people are looking very carefully to see how long does the immune response, how good of an immune response do you make? How long does it last? Does it protect you? And we sort of know about 6 months worth of this data that says that the immune responses seem to last for 6 months. So there's obviously a lot more, a longer way to follow these patients. The other thing that we're all worried about are these variants, right? There are variants in the UK, variants in South Africa, there are variants in California. We have a whole lab, I didn't include any slides, but Brian Pining in the molecular genomics lab is looking for variants in Oregon all the time. When people have COVID found, it also goes to his lab so that their genetic code is sequenced, and we can see if the virus is changing, because the virus could change and make our tests out of date and no longer useful. And more importantly, it could change in a way that makes it less susceptible to therapy or resistant to vaccination. And while there are modest reductions in the sensitivity of these viruses to our vaccine responses, they all seem to be susceptible to the immune responses that are provided by Pfizer and Moderna. And then there's a question of, will the vaccines work for everybody? Are there going to be patients like the elderly and immunosuppressed who don't make great immune responses? Will they be protected? So this led us to begin our own little vaccine trial, acknowledging that it was more complicated than others because we wanted to be ready with a second line vaccine in case the first line approaches weren't protective for everybody. And so what we've done is we're using DNA instead of RNA. It's another nucleic acid. It's the actual gene for the spike protein. It gets injected, and then it has to go to the cell and make the message and the proteins and the immune response. So this is what we've been doing. We've probably got 12 or so patients vaccinated with this to try and assess their immune response, but we don't have the data yet to share. But this was also a study funded and sponsored by a company, Oncosec, but also funding was provided by our philanthropic community up here in Portland. The other thing, it was interesting, we had a scientist whose wife had a baby and got vaccinated after the baby was born, and she's a physician as well. And we're interested to see if these antibodies that are produced against the vaccine could also be found in the baby's milk. And lo and behold, if you look over here, before vaccination, there were no antibodies in the milk, and then after vaccination, there are two different kinds of antibodies that are important and protective and could help lead to protection of the newborn were produced. And this actually small study with 6 patients generated lots of national interest, including a write-up in Forbes magazine, which was very exciting for everyone involved. And so I'll finish with just saying that this timeline was amazingly quick. I want to recognize what here are called the silent unrewarded lifesavers, which are the lab researchers all across the globe who did all this work and have risked their personal safety and worked very hard to help find effective therapies, but most importantly have worked on these vaccines, which in an unprecedented time, in less than a year, we have a vaccine that works. And we actually know the people who developed the vaccines because their technology was first developed for cancer vaccines. And we've actually given dozens of these cancer vaccines to patients before they turned their attention to coronavirus. And it must be pretty special for them to lie awake at night and think about how they've saved millions of lives by developing this vaccine. So I will stop there, and I'm happy to answer any questions. Thank you very much.
Well, thank you, Walter. Great presentation and very helpful. There are a number of questions. I'll first start with the disease itself, those who have had COVID. And could you talk a little bit about what's now being called the COVID long haulers and the treatment of the residual illnesses and what we're seeing? It's a small population, I understand, but nonetheless, one that we hear about frequently.
Yes, and they present quite a challenge because they no longer have the virus, but they are complaining of and experiencing a variety of nonspecific symptoms. Then they can affect their quality of life, their ability to work. I know there are lots of registries being made, and the NIH is focusing on what do we do for these patients. Is this a consequence of an immune response they had to the virus? Is it a consequence of what damage the virus did when it infected some of these tissues? I don't think we have good answers, and for the most part, and you may know more about this, Tom. For the most part, it's supportive care, trying to just help them get through whatever symptoms they're having and see if they don't get better over time.
You know, and as I look at your initial slides, I think we're just now at the start of understanding it. You know, another year or two down the line, we'll look at today and say, here's the number of long haulers. And then going forward, as we consolidate data, consolidate the groups, gain more experience, we'll have more to say. But I think to your point, we're just at the very beginning of understanding it, getting the groups together, and identifying things that work and don't work. But at the moment, probably not a specific treatment for those long haulers, but hopefully in the future we'll have something. So back to the vaccine, for those people that are hesitant about getting the vaccine, what would you say is your best advice to help people embrace the idea of getting vaccinated?
That's of course a great question, and I've been reading a lot about different people's approaches to this, because being a physician and a scientist, my first stop would be better education. We need to educate better, and I know that one of the fears was, well, Operation Warp Speed. That means we're rushing. And we're not paying attention to the rules of the road, and we don't really know everything we need to know. And so people were worried that this was being pushed through at such a rapid rate that somehow important safety steps were being missed. And I can say for a fact that that's not the case. The reason we were ready in a year, I alluded to, was that we already knew about the safety of these vaccines and how to make them in terms of their use for cancer patients. And so within a month of the first viral infection in Wuhan, they had a sequence of the virus. They knew what the messenger RNA was for the spike protein, and they put it into their lipid particle. And so all the right steps were done. The proper studies of vaccinating people and not vaccinating others, comparing safety. There was no government fiat decision to go ahead and approve this. It went through all the appropriate steps for the FDA approval, independent advisory council, then FDA review. And so from an educational standpoint, so try to share that this went through in record time, but they didn't skip any of the steps they used to use. That you have to acknowledge that we don't know about long-term side effects. Could there be long-term side effects? There could be. They're not very common, they're not very frequent, but the short time span in which this was developed makes it impossible for us to really comment on what the long-term side effects could be. But there are other reasons than just the science that people don't want to get vaccines. It's got to do with trust of the healthcare community, trust of the government, and those are bigger challenges for all of us. My first thing is always to try and lead by example. So I got in line as soon as I could to get my vaccine. That may help some people make their decision, but it won't help everyone. But it's important that we get enough of us vaccinated so we get that so-called herd immunity level, and that our lives can return even more to normal.
Talk a little bit about vaccination in children, and where are we in that space?
Yeah, so children have, of course, they've gotten the lowest priority here, and that's probably a proper decision, because they're the least affected, and they're the ones who can tolerate COVID infection the best. So the early studies that were done with vaccines did not include children. So we don't know anything about safety in children, and so current recommendations are not vaccinating children. However, both Moderna and Pfizer have studies ongoing, and I expect that we'll see data anytime in the next few weeks or few months where I'm trying to remember the lower age limit, but certainly they're looking at adolescents, certainly high school kids, and looking younger to see if those vaccines also will be effective and protect children.
I think the initial data on the Pfizer vaccine was, it appears to be protective without side effects, 12 to 16.
Yeah.
And the thought is that age group could get, I think, vaccinated towards the end of the summer. and the data for 6 and above are still ongoing and out there. Talk a little bit about if you are vaccinated, can you still get COVID and can you still transmit COVID? What's the thought on how cured are we?
Yeah, and I think the answer, no vaccine works 100% of the time, right? We don't have a test to measure the level of an immune response. So there's no way we're going to know exactly out of the couple 100 million people who get vaccinated whose immune responses reach the level that are protective. We don't even know what those levels are right now. So I suppose you'd have to say that it is theoretically possible that one could get infected again. There are certainly instances of people who get COVID who do get COVID a second time. And it's been proven by looking at the virus that the virus they got the second time is different than the virus they got the first time. Now, that could be because their immune response wasn't as strong. And we actually know that the immune responses that people make against the Pfizer and Moderna vaccines are of a higher magnitude than they are after a natural infection. So vaccinated patients may also be better protected. But then there's this whole thing about variants, right? And so it is totally possible, and we have data to suggest it is so, that these variants, and a lot of the variants change the spike protein, which is the target we're using to prevent or get rid of the coronavirus. They change that spike protein, and it's possible that I might make a great immune response against the UK variant that doesn't work against the South American variant. We're not finding it to be a total black and white issue. It looks like the variants are more resistant to the responses that are produced by a vaccine against a different strain, but the level of antibodies is still plenty high enough that it should prevent us infection. So I feel quite safe, not perfectly safe. Like even I'm not perfectly safe when I cross the street either, right? So, and there was a second part, Tom, to that.
I was just gonna add that the OHA a week or so released that there were 168 known cases of patients becoming COVID positive after being fully vaccinated. But that represents a point, I think it was either 0.0006 or 0.00006% chance. So it is statistically possible, and to your point, nothing's perfect, but this is really, 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 really good.
Really, really effective. And it depends on when you look, right? Because if you're vaccinated, these mRNA vaccinations require 2, and optimal protection occurs after 2. And you can see it probably takes 14 days for those lines I showed about infection. 12 to 14 days, you start seeing the difference between placebo and vaccination. So if you're infected early after your vaccine, before you made an immune response, that wouldn't be a surprise. But your point is well taken.
And there's a question in the question and answer that I don't know if you can answer or not, but with the emergency use approval process, at what point will the FDA say it's approved, and what will that mean? Is there a timeline for that?
I don't know. I mean, there's clearly the next step is based on, and when you have an EUA, an emergency approval, you are required to keep track of where the vaccines went, who got it, what the side effects were, what the efficacy was, and then you put together a full biological licensing application that goes to the FDA, and the review is even more detailed than the one was for the EUA. And then the big deal at the end of that is that then it's approved and it changes billing and all those other things. But I can't imagine that it will be long before both of those vaccines go through that process.
So which is better, J&J, Pfizer, Moderna? There's a you hear different statistics.
Yeah.
What's your thought on that?
Well, the real answer is I don't know. I am totally comfortable that there's no difference between the Moderna and the Pfizer, the mRNA vaccines. There's, you can't, acknowledging they've never been compared head to head, which is the appropriate way to do it, is to compare both vaccines in the same study. I don't think we're ever going to see that. But if you look at the protection, the immune responses, the side effects, I'm convinced that they're the same. And Pfizer, Moderna, one half dozen of the other. The viral vaccines are a little different. Johnson & Johnson, it's a one-shot vaccine for many. Sometimes the data looks like it's a little less effective when you look at the numbers, but they're still within the realm of they're having similar efficacy results, and the side effects are not terribly different. They're AstraZeneca, I'm not so sure, because they're and I'm glad I don't have to choose between getting that vaccine. My answer to people is, well, which vaccine should I take? And the answer is whichever one you can get, right? That's how I would answer that now. We don't know that one's superior than the other, but we know that it's really superior to be vaccinated than to be unvaccinated. Little scary stuff with the AstraZeneca vaccine with these hematologic disorders where you have to look at this. Anytime you're vaccinating hundreds of thousands of people, some of them are going to develop blood clots, right? So sometimes it's hard to figure out, well, what's really normal and what would have been expected to have happened anyway. But these are very unusual blood clots in the brain and other places. They're small numbers, right? You can still write a paper with 5 patients in it, and it can go in arguably the most famous journal in the world. But they're a little scary. So I'd be skeptical about right now AstraZeneca until we get more data. And I'm glad we in the U.S. aren't faced with having to say, well, gee, AZ is the only one we've got. I think it probably still makes sense to get vaccinated because the incidence of these side effects is so rare. But rare for a population if it's your family, if it's your person. So I don't know if you saw it, but I was kind of impressed with the wife of one of the patients that died from England, I think it was England, who got on TV and said, yes, I know my husband died. It's a terrible thing, but you should all still get a vaccine because the chances of this happening are so much lower than the chances of something bad happening with COVID.
I think for the chamber to understand, the AstraZeneca vaccine has not been given emergency use in the United States. And if all vaccines are delivered, the Moderna, Pfizer, and the J&J will more than cover the U.S. population. And then one other thing is in terms of herd immunity, that I think it's important that we vaccinate. It's not just herd immunity in the United States. If we don't vaccinate the rest of the world will develop new variants that will make it more challenging for our herd immunity to stick. So I think it's really important that we actively pursue and support vaccination all around the world, because that's not an academic issue. It will come back to bite us, if you will, if we don't vaccinate, because the variants will then come back and we can see another wave. So I just kind of make that point.
It is in our self-interest to share or use whatever vaccines are left over to get to other folks around the world, without a doubt.
So this question is for Chris. How's the hospital doing in Medford, and what's your report from the caregivers there?
Yeah, Tom, I could sit here and talk to or listen to Dr. Erber all day. Appreciate your knowledge and sharing everything that you've shared with us today. As far as the hospital, today I'm happy to report we have no COVID patients in-house at this time. And in terms of preparing for a surgeon, we've done a lot of work. Certainly at the beginning of this, when you saw the graph that Dr. Erber shared earlier, when we were seeing the surges starting to occur, we worked very closely with our regional and system partners to identify the things that we needed to do in preparation for a surge, making sure that we have the equipment, the supplies, all of the things, the workflows that are needed to ensure safety of patients and our caregivers. And so done a lot of work on that end, on the front end of this, and I could say that we're in a good position right now. Certainly have no concerns at this time for PPE and other equipment that's needed. We have plans in place to surge up to twice the capacity that we currently have right now in the hospital. So I would say, we're in good shape. We're again, as more vaccinations are being done, and we encourage that to continue, we'll see, less and less of incidences of these cases in our community.
Dr. Erber, there's a question in the chat area. What about pregnant women? What's the recommendation for pregnant women?
I think that's have a one-on-one conversation with your doctor, but I think most people recommend that they get vaccinated. I think that's a CDC, NIH recommendation. Yes.
And then are people with anaphylaxis, should they get the vaccination? Is a history of anaphylaxis, I suppose a peanut allergy here, is that a reason not? And maybe a broader question, is there any recommendation of when you would not get the vaccine, something that's hard and fast, and of when you would not get vaccinated?
Yeah. Anaphylaxis is very, very rare occurrence. The recommendations have been if you have had an anaphylactic reaction to others, and these anaphylactic reactions aren't just like the hives and things like that. Anaphylactic reactions, if you are sick enough and you get taken to the ER and you need to be given epinephrine and your airways were closed. They're looking for what the common thing might be among the vaccines that would trigger this thing, and there's nothing really known. And to use the phrase that we've heard so much the past year, out of an abundance of caution, the recommendation has been to sort of tread lightly where people have had a reaction before. I don't think it should automatically disqualify you. I don't know of any other reactions that would. My own view would be tempered by whether you're a nursing home patient at high risk or someone overweight and elderly versus you're a 16-year-old player in the band who's got one of these allergies. That might affect my individual decision.
So I think what I'm hearing throughout this, the thread is get vaccinated. And if you have any questions, talk to the doctor and likely get vaccinated. Having said that, Scott Kelly in the question and answer asked, how many Providence employees have been vaccinated, and it's over 50%. That's good news that it's over 50%. It's higher in the Portland area in our caregivers there, but it's at the same time disappointing that it's only above 50%. So I think there's a lot of education, but as we understand more and as more people get vaccinated, and as we continue to see the low volume of negative reactions and the high probability of a positive outcome, that the evidence is more and more certain that vaccination is in your best interest. And so just continue to encourage people, if you haven't been vaccinated, to reconsider it, get as much information. But the information to date that we in the medical community have is that get yourself vaccinated. So I think that's, let me just look at the list one more time. How, I get, there's a specific questions, how long do the antibodies, the serum tests remain positive after infection? So I guess there are two questions in there. If you had it in the, last March, how long are you vaccinated, or can you resist the infection? And then how long do you see a vaccination working? Will it look like the yearly flu vaccine, or is this going to be more like a mumps rubella kind of vaccine?
Yeah. So answers to both are I would have to say unknown to state specifically, but we do the coronaviruses make up a family, and we've dealt with a few. The first SARS killed 3,000 people. The MERS, which is mainly a Middle Eastern virus, killed a large number of people, and it's still around. But there are other coronaviruses that have the same family that cause the common cold. And what we know from them is that you get reinfected each year with some of those coronaviruses. And we know that the antibody responses to some of those don't last very long. They don't last years. But we do know, example, for other serious infections that we get and we get vaccinated against, things like tetanus and things like that, our antibodies can last for years when you get vaccinated against those. So there's data that the coronavirus family, the immune responses are not going to last for years. We also know that in people who have had COVID, some of them 6 months later don't have any antibodies in them. Antibodies get produced within a week or so, one kind, and it can go away, and then one kind we call IgG tend to hang around a lot longer, but we know that they're not staying present forever. And I showed that one slide, which is measuring antibody responses. And since we've only been vaccinating people at the longest for 6 months or so, the longest data we have says that 6 months there's still antibody there. And in most of the patients, whether it's there at 9 months and 12 months is something we're going to find out over the next 6 months.
Well, we're at time. Dr. Erba, I want to thank you for your service, the research over the last year, and this talk. I believe it's topical information and well received by the group here. So Lydia, do you have, I'm wondering what the next step here is in terms of bringing this meeting to the end. But I want to thank you, Walter, and thank you. The Chamber for hosting us today.